Assalamualaikum and a very good day to our lecturer Dr. Fidaus. We are from Group 8 and today we would like to present on Bacillus cereus, a microbial pathogen that commonly associated with food. Before we begin our presentation, I would like to introduce our group team. Our team consists of 10 people. They are INTAN SYUHADAH, SHASHNEETA, NAJIHAH DANIA, SYAHIRA SYAFIQAH, SITI ZULAIKHA, NURUL FATIMAH AISYAH, Fatin Nur Nabila, HARUL NATASHA, SITI NURUL AFIQAH, and ZULAIKHA. Next, I will briefly explain the structure of our presentation. Our presentation is divided into 5 main parts. First, we will begin with an introduction to Bacillus cereus. Second, we will discuss its toxic pathogenic metabolites. Third, we will explain the factors affecting toxin production. Next, we will describe the mechanism of infection. And lastly, we will conclude with foodborne disease caused by Bacillus cereus. Assalamualaikum. My name is SYAHIRA SYAFIQAH binti Muhammad Shafi'i. My matrix number is A201258. Today, I would like to present on Bacillus cereus, a bacterium that is significant in microbiology and food safety. In terms of nomenclature, this bacterium is scientifically known as Bacillus cereus and was first described by Franklin and Franklin in 1887. It is commonly referred to as B. cereus in scientific literature. Regarding classification, Bacillus cereus belongs to the domain Bacteria, phylum Firmicutes, class Bacilli, order Bacillus, and family Bacilliaceae. It is under the genus Bacillus with its own species designation as Bacillus cereus. Now, I will explain the characteristic of Bacillus cereus. In terms of morphology, Bacillus cereus is a rod-shaped bacterium that appear purple or violet under gram staining, indicating that it is a gram-positive bacteria. The size is approximately 1 to 1.5 micrometer in width and 3 to 5 micrometers in length. It is capable of forming endospores, which are highly resistant structure, allowing survival under heat, desiccation, and chemical exposure. The bacterium is also motile, with flagella distributed around its surface. For colony appearance, Bacillus cereus typically forms large, flat, irregular, and opaque colonies on nutrient agar, sometimes resembling frosted glass. From a physiological and biochemical perspective, it is a facultative anaerobe, meaning it can grow both in presence and absence of oxygen. It is a catalase positive, indicate its ability to break down hydrogen peroxide into water and oxygen, but oxidase negative, showing that it does not produce cytochrome oxidase. The bacterium can ferment glucose and produce acid and gas and secrete various extracellular enzymes, including protease, lipase, and lecithinase, which play important role in nutrient breakdown and its pathogenic potential.
Assalamualaikum. My name is Siti Nurul AFIQAH binti ABDUL and my matrix number is A200697. I will continue for the introduction part. So, where we can find Bacillus cereus? Bacillus cereus is a gram-positive and spore-forming bacterium that is commonly found in soil, water, and vegetation as reported by Comda et al. In 2025. It is also present in the roots and tubers of starch-rich plants such as cereals and rice, which allows it to enter the food chain during harvesting and food processing. According to Comda et al. In 2025, ready-to-eat products, soups, sauces, dairy products, and vegetables are the high-risk food. Additionally, cooked rice are the most commonly affected foods. This is because Bacillus cereus can produce heat-resistant spores that survive normal cooking processes. Calvigioni et al. In 2023 also reported that the bacterium can persist in food preparation and processing environments by forming spores and biofilms, which allows it to survive harsh conditions. Now, I will explain the history of Bacillus cereus in food safety. Bacillus cereus has been recognized as a foodborne pathogen since the mid-20th century. Stanford's Arneson et al. In 2008 reported that it was first linked to diarrheal outbreaks in Norwegian hospitals between 1947 and 1949. The emetic syndrome was later identified in the early 1970s after several food poisoning outbreaks happened in the United Kingdom. and it is linked to the consumption of cooked rice that already contain the toxin called cerulite. Next, according to ABDUL and Pavoni, in 2025, Bacillus cereus is responsible for approximately 1.4 to 12% of global foodborne outbreaks. Comda et al. In 2025 also highlighted that in Europe, it ranked as the second most common outbreak agent in France and the third most common in the European Union. This shows that Bacillus cereus remains an important issue in food safety and surveillance.
Assalamualaikum everyone, my name is FATIMAH HARUN, metric number A201252. For this session, I will explain the enterotoxin produced by Bacillus cereus. Bacillus cereus produce enterotoxin that are responsible for diarrhea-type food poisoning. These toxins mainly act on the intestinal epithelial cell and cause fluid loss in intestine. There are three main enterotoxins, which are hemolysis BL, no-hemolytic enterotoxin, and cytotoxin K. First, hemolysis BL or HBl. HBl is three-component pore-forming toxin. It consists of one binding component, which is B, and two lytic components, L1 and L2. This toxin is heat lipoprotein complex with molecular weight of about 100 kilodalton and also sensitive to proteolytic enzyme. HBl acts by forming pore in intestinal epithelial cell membrane. The toxin component assemble on the cell surface and create pore. This causes osmotic imbalance, which allow food to leak into the intestinal lumen. As a result, patients experience watery diarrhea. The main effect of HBl include water diarrhea, abdominal cramp, and nausea, and these symptoms usually appear 6 to 50 hours after injection. Next, no-hemolytic enterotoxin or NHE. NHE is three-protein complex made up of NHEA, B, and C. Similar to HBl, it is a heat lipoprotein toxin with molecular weight of around 100 kilodalton, and it acts on intestinal epithelial cell. NHE form pore in cell membrane which increase permeability to ions such as sodium, potassium, and chloride. When these ions move out of the cell, water followed by osmosis. This leads to fluid circulation into the intestine. Because of this mechanism, NHE causes water diarrhea, abdominal pain, with symptoms appear 6 to 50 hours after injection. Lastly, cytotoxin K. Cytotoxin K is single component beta barrier pore-forming toxin. It's also heat labile but smaller in size, with molecular weight of about 34 kilodalton. Cytotoxin K mainly acts on the intestinal epithelium, but in severe cases, it may affect other tissue. Compared to hemolysis BL and no-hemolytic enterotoxin, cytotoxin K is highly cytotoxic. It inserts into cell membrane and forms stable pore, which lead to cell lysis, epithelial damage, and severe fluid loss. Clinically, cytotoxin K causes severe diarrhea and strong abdominal cramp and has been associated with serious or even fatal outbreak. In summary, these three enterotoxins damage intestinal cells through pore formation, which lead into the fluid loss and diarrhea symptoms in Bacillus cereus food poisoning. Thank you.
Assalamualaikum. My name is NURUL DANIA. My metric number is A203967. I will explain the emetic toxin produced by Bacillus cereus, which is known as cerulite. This toxin is responsible for the vomiting type of food poisoning and is very different from the diarrheal enterotoxins. Cerulite is a cyclic dodecapeptide, meaning it forms a closed ring composed of 12 alternating amino acids and oxy acids. This unique structure makes the toxin highly stable and resistant to degradation. In addition, cerulite is lipophilic, allowing it to interact easily with lipid membranes and making it difficult to remove from contaminated food. One key characteristic of cerulite is its extreme heat stability. It remains active even after exposure to 121 degrees Celsius, meaning reheating or cooking contaminated food does not destroy the toxin. This is in contrast to Bacillus cereus enterotoxin such as HBl and HE, which are heat labile and easily inactivated. At the molecular level, cerulite acts as a potassium ionophore. It transports potassium ions across mitochondrial membranes, disrupting the mitochondrial membrane potential and reducing ATP production. This interference with cellular energy metabolism explains the strong toxic effect of cerulite. In conclusion, the unique chemical structure, high thermal stability, and specific molecular mechanism of cerulite make it an important pathogenic metabolite of Bacillus cereus and a significant concern in food safety.
Environment factors influencing toxin production can be divided into intrinsic and extrinsic factor. Intrinsic factors, which are properties of the food or environmental itself. pH is one of the most critical factor, where a neutral to slightly acidic pH, which is 4.5 to 7, can enhance the toxin stability and activity. In this pH range, toxins such as emetic and enterotoxins remain stable and biologically active. Extremely acidic or alkaline conditions can inhibit the growth and reduce the toxin production as well. The next is water activity, which is another important intrinsic factor, where high water activity supports bacterial growth and continuous toxin synthesis. However, when the activity decreases, it can trigger stress responses such as sporulation, but toxin production may still occur. The last intrinsic factor is nutrient availability, where carbon and nitrogen sources influences metabolic activity, and certain substrates, especially simple sugars, promotes toxin production more effectively than others. On to extrinsic factor, which are external conditions surrounding the bacteria. Firstly, temperature remains a dominant influence, where temperature within 28 to 37 degrees Celsius increases the bacterial metabolism and toxin synthesis, while low temperature slows down the toxin synthesis. And next is relative humidity, indirectly affects the toxin production by altering water activity. High humidity environment tend to support bacterial growth and toxin accumulation, especially in stored or cooked food. Finally, oxygen availability as an extrinsic factor. Bacillus cereus is a facultative anaerobe, meaning it can grow or produce toxins in both the presence and absence of oxygen. But however, changes in oxygen level can influence the type and amount of toxin produced by activating different metabolic pathways.
Next, I will explain how the growth curve of Bacillus cereus relates to the toxin production, with a focus on why the toxin is produced at the specific stages of the bacterial growth. First, this diagram shows the typical bacterial growth curve, which consists of the several phases, which is the lag phase, the exponential phase, or we call it as a log phase, and the stationary phase. The production of toxins, specifically cerulite, is closely linked to the changes in the bacterial metabolism across these phases. Starting with the lag phase, the bacterial cells are adapting to a new environment. During this phase, there is little to no cell division, as the new bacteria are synthesizing essential enzyme and adjusting their metabolic pathway to suit the surrounding conditions. Because the cells are focused on adaptation and survival, the toxin production does not occur at these stages. At this stage, producing toxin would be energetically inefficient when the cells are not yet actively growing. Next is the exponential or log phase, where the bacteria begin to divide rapidly. During the early part of this phase, the cell prioritizes biomass productions and energy generation to support their rapid growth. As the result, the toxin production is still minimal or absent. However, as the culture progresses into the mid to late exponential phase, cell density increase significantly. At this point, quorum sensing mechanism become activated. This allows the bacteria to sense that there are enough population size has been reached, which triggers the expression of genes involved in virulence and toxin production. This leads into the early stationary phase, which the main stage where toxin production occur. During this phase, nutrients in the environment become limited and waste products begin to accumulate. As a growth slows, and the cell division stabilize, Bacillus cereus shift from growth focus strategy to the survival focus strategy. Toxin production at these stages provide a competitive advantage as the toxin can damage uh, toxin can damage the host cells or suppress competing microorganism, helping the bacteria access additional nutrients. Furthermore, toxin synthesis is considered as a secondary metabolic process which typically occurs when active growth is no longer the primary priority. Uh, Finally, during the late stationary phase, the number of viable cells remains relatively uh, constant or begins to decline. At this point, the toxins level actually reach a plateau, meaning that the production stabilize or slightly decrease. The toxin production stabilize and slightly decrease. The bacteria are now conserving energy and preparing for the long-term survival rather than producing the additional toxin. In conclusions, toxin productions in Bacillus cereus does not occur randomly. It is tightly regulated and mainly associated with the transition from the exponential phase to the stationary phase. This timing reflects a strategic shift from the rapid growth to survival and competitiveness under the stressful environment conditions.
My name is Siti ZULAIKHA and I'll be continuing my part on how pathogens contaminate the food. Bacillus cereus is a versatile pathogen widely known for causing fried rice syndrome. This bacterium is gram-positive and found almost everywhere in the environment, particularly in soil. Because it lives in the soil, it easily contaminates plant-based food like rice, vegetables, and spices. The key to its persistence is ability to form endospores. These spores are incredibly tough. They can survive standard cooking temperatures that will kill other bacteria. Additionally, in food processing plants, Bacillus cereus can form biofilm on stainless steel equipment, making it difficult to clean and serving as a continuous source of contamination. So, how does it get into our system? The route of entry is strictly oral. We have to ingest the food. The transmission is driven almost entirely by what we call temperature abuse. Here is the classic scenario. You cook a large pot of rice. The heat kills the active bacteria, but the spores survive. If that rice is left out at room temperature for several hours, those spores wake up. They germinate into vegetative cells and begin multiplying rapidly. By the time you eat the leftovers, the food is teeming with bacteria or the toxins they have already produced. Next is mechanism of disease. I will begin with the emetic syndrome, which causes vomiting. It is important to define this as an intoxication. This means the illness is caused by a chemical poison, not by an active infection. The damage comes from a toxin that is already present in the food before we eat it. The specific toxin responsible is called cerulite. Its structure is distinct because it is cyclic peptide, essentially a closed ring of amino acids. This ring shape is chemically locked, which makes the toxin incredibly stable. It can survive high cooking temperatures, stomach acid, and even our digestive enzyme. So, how does it affect the body? First, because cerulite attracts fats, it is absorbed very quickly from the digestive tract into the bloodstream. Once inside, it disrupts our cells in two ways. First is by the mitochondrial attack. It acts as an potassium ionophore. This means it inserts itself into the mitochondria and moves potassium ions across the membrane. This destroys the electrical balance and shut down the cell's energy production. Second is the neural trigger. Most importantly, the toxin targets the nervous system. It binds to 5-HT3 receptors, located on the vagus nerve in the stomach. This sends an immediate distress signal to the brain vomiting center. Because the toxin is preformed, this reaction is very fast, often occurring within 30 minutes. Next, we have the diarrheal syndrome. This operates differently as a toxic infection. In this case, the bacteria must survive the stomach, grow inside the intestine, and produce toxin directly within the host. The bacteria do not produce this toxin immediately. They use a communication system called quorum sensing to detect their population density. So how does the quorum sensing work? Is by they secreting a signal peptide called PAPR. When the bacterial population is high, this signal accumulates and activates the master switch called PLCR. Once activated, PLCR turns on the genes to produce enterotoxin. The bacteria release three main toxin, which is hemolysin BL, non-hemolytic enterotoxin, and cytotoxin K. We can describe this as a pore-forming toxin. So the process occurs in specific steps. First is addition. The bacterial cell attached to the lining of the small intestine. Second is assembly. The toxin components assemble on the surface of the host cell. For example, one component acts as a docking station to bind to the cell. So third is pore formation. Once assembled, the toxin complex create a physical pore or channel through the cell membrane. So this pore disrupt the cell balance. Water rushes into the cell due to osmotic pressure, causing the cell to swell and burst. This destruction of the intestinal lining leads to fluid leakage and watery diarrhea.
To understand Bacillus cereus food poisoning, we first have to look at the numbers. The dose required to make someone sick depends on the type of syndrome. For the diarrhea type, which is a toxical infection, a person typically needs to ingest about 10 to the power of 5 bacterial cells per gram of food. However, high potency strains can cause illness with as little as 10 to the power of 3 cells. On the other hand, the emetic or vomiting type is a pure intoxication. It is caused by a preformed toxin called cerulite. Humans are very sensitive to this. Health risks begin at just 1.8 micrograms per kilogram of body weight. Interestingly, if a strain produces a variant called isocerulite A, it becomes 8 times more toxic, meaning even smaller amounts can cause severe illness. For colonization and absorption, once the bacteria or toxins are inside the body, their journey differs. Diarrheal bacteria face a tough environment, only about 26% survive the journey through the stomach. To protect themselves, they anchor directly to the gut lining to release their toxin, which keeps toxin from being destroyed by digestive juices. The vomiting toxin cerulite is much steadier. It is completely unaffected by stomach acid or enzymes. Instead of staying in the gut, it uses the intestine as a gateway to enter the bloodstream. From there, it travels throughout the body, building up in the liver, spleen, brain, and fatty tissues. The body eventually clears it out through fecal excretion. Let's look at how this toxin actually destroys cells. The enterotoxins, the ones causing diarrhea, work like a construction crew. They bind to the cell surface in a specific order, first part B, L1, and then L2. Together, they form a lytic pore or hole in the cell membrane. This causes potassium to leak out, the cell to swell with water, and eventually burst. Cerulite works differently by attacking the cell's energy source, the mitochondria. It acts as a potassium carrier that destroys the mitochondria's electrical charge. Without this charge, the cell cannot produce ATP or energy. This energy failure can lead to tissue death in the liver and kill pancreatic beta cells, which stops the body's ability to produce insulin. Finally, how does the body respond to this damage? In the diarrheal version, the loss of potassium triggers a major immune response called the NLRP3 inflammasome. This causes the release of pro-inflammatory chemicals like interleukin-1 beta and interleukin-18, leading to a form of cell death called pyroptosis. This is why we see the classic symptoms of painful cramps and watery diarrhea. The vomiting response is more about nerve signaling. Cerulite targets 5-HT3 receptors in the gut, which send a signal to the vagus nerve to the endoplasmic reticulum. Beyond emesis, it also causes internal cell stress and can cross the blood-brain barrier. This explains why some severe cases involve not just stomach issues, but neurological complications like seizures or convulsions.
Assalamualaikum. My name is INTAN SYUHADAH with matrix number A200307. I will continue this presentation with foodborne disease. Bacillus cereus gastroenteritis, or its common name, fried rice syndrome, is a common occurrence. This disease have two very different clinical syndromes. The first is the emetic syndrome. The bacteria produce toxins inside the food. When the food is consumed, the unlucky person will get food poisoning. This toxin is called cereulite. This can occur during its incubation period, which is quite fast, from 30 minutes until 6 hours. In a real-life scenario, if you accidentally consume contaminated pasta or rice, you will start feeling sick in a short period of time after you have consumed it. The clinical symptoms are dominated by acute nausea and projectile vomiting. This is an indicator that the body is trying to get rid of the toxin out immediately. The person will feel a lot better within 24 hours. The second symptom is diarrheal syndrome. This is different from the previous syndrome because it is a toxico-infection. The person will not only consume the food, but they will also consume the live bacteria inside the food. The bacteria will enter the body and produce toxins inside the small intestine. The incubation period will be much longer, can be up to 6 or 15 hours, and take a full day. The clinical symptoms that can happen are profuse watery diarrhea and painful abdominal cramps. Unlike emetic type, vomiting is quite rare for diarrheal syndromes. Thank you.
I'm ZULAIKHA Muhammad Zamasari. I will continue the presentation. Next, let's look at the type of food that are commonly associated with Bacillus cereus food poisoning. The emetic syndrome is usually associated with starchy food, especially cooked rice, potatoes, pasta, and pastry that are left at room temperature for too long. A common example is fried rice syndrome. Meanwhile, the diarrhea syndrome is associated with meat products, dairy products, soup, sauces, and vegetables. This food provides suitable condition for Bacillus cereus spore to survive cooking and grow if the food is improperly stored. In terms of population at risk, Bacillus cereus can affect anyone, including healthy individuals. However, the risk is higher among infants, young children, the elderly, and also immunocompromised persons. People who consume food from cafeterias, schools, and hospitals are also more vulnerable, especially when food is prepared in large quantities and stored improperly. Prevention and control of Bacillus cereus focus on proper food handling and storage. Cooked food should not be left at room temperature for a long period of time and should be refrigerated promptly at temperatures below 4 degrees Celsius. Reheating food thoroughly before consumption is also important to reduce the risk of infection. Good personal hygiene, clean food preparation surfaces, and adherence to food safety practices such as HACCP are essential in preventing Bacillus cereus contamination. That's all from me. Thank you.